but I did not get the time to send the
Okay, ma'am.
copy. We'll send it today. Right. Now let's start with Phylum Porifera. We are going to just have a look at the general characteristics, their classification, and we are going to have one type study, that is Icon, and then we are going to see the water vascular system in the poriferates. So when you come to see Porifera, you just know them as sponges. And then you're going to have a detailed study about one particular organism, that is Icon, its external and internal structure, what are the different various types of cells that are present, what is the skeleton of the sponge made up of, what is the canal system, then the physiology of Icon. Then how the the water vascular system, what is there, or the water canal system, what is there, it is the most unique feature of sponges. So these are the I request you all to mute yourselves. That is the only thing I'm asking you. Please mute yourselves. Yes. Oh, wait. Right. So let me start sharing my screen. There are two particular PowerPoints that I just kind of took it from the internet. In fact, couldn't get the time to get.
తాళం లోపలనే పెట్టి ఇట్లా తాళం మొత్తం వచ్చినావా వేసి వచ్చినావా
మీరు తాళం తర్వాత పెట్టుకోండి. Can you please mute yourself? So, right. So there are two different kinds of what do you call it, PowerPoints that are present with me right now. I have taken it from the internet. In fact, nothing original as of now. So when it comes to Phylum Porifera, you see that they are the multicellular sponges. In fact, most primitive multicellular organisms, and excepting the family of freshwater sponges, which is Spongelidae, all of them are exclusively marine organisms. Icon is a calcareous sponge with an internal skeleton which is composed of calcium carbonate spicules, and the typical example which is showing all the features in this particular phylum, in fact. So when it comes to the general characteristics, Phylum Porifera begins the evolution of multicellular organisms or multicellular animals. Body organization is higher than that of the protozoans. But when compared to other metazoa, metazoans, you get to see that they are quite primitive. So only organization is better than protozoa, but not as good as the rest of the metazoans. So the systematic position of sponges has always been doubtful as to should they put them in the plant kingdom, or should they put them, because they're like sessile plants, do not have feet or locomotory or organelles or organs for that matter. So where should you put sponges? So with that, what they have done, they have studied the histology, the cytology, the embryology of the sponges. This confirmed that they have a very low level of organization, but it is there. As a result, they gave a separate status and a separate phylum to it and called them as parazoans. So Phylum Porifera has around 9,000 species, of which almost 4,000 species are extinct as of now. So when it comes to the general features of Phylum Porifera, you can get to see that they are multicellular bodies with pores, that is, where water and canals and chambers are there. So this is known as a water canal system. Water currents play a very important role in this, and there is a radial symmetry present, or it can even be asymmetrical. No true organs or no organs are present. No tissue level of organization is present. Digestion is mostly intracellular, excretion and respiration is by diffusion. And then freshwater sponges are they belong to Spongelidae. Body is either vase or cylindrical or globular. They can exist in solitary ways, or they can be colonial also. All the members are multicellular and diploblastic. That is, they have two layers in the body basically, as there is a picture. So they have a two-layered body basically. One is made up of pinacoderm. This is the pinacoderm. This is the choanoderm. That's all. Body has two layers, so it is known as diploblastic. So pinacoderm is the non-flagellated dermal layer, but the choanoderm has flagella to it. So it is known as the flagellated layer or the choanoderm. and it has flagellated cells which are known as Quinocytes. So this is the characteristic feature of phylum Porifera. Now there is a gelatinous mesoglia or mesenchyme which is present between the dermal and the flattened layers, and it has numerous different kind of amoeboid cells in the middle, in this middle basically, and numerous there are a large number of pores, chambers, canals that are present throughout the body. Do I have a picture regarding that? Let us see over here itself. See, if you see, yeah, all this basically is made up of pores, holes, canals, etc. Numerous small openings called as ostia are found all over the body, and there is a large central opening which is known as an osculum. These are the ostia that are present. This is the osculum. Now, the central body cavity is known as this one is known as the spongiocele or the paragastric cavity, and body is made up of a skeleton of rods which are known as spicules. See over here, you see this one. Spicules can be either made up of calcium or silica, or the fibers which are made up of spongin. So these hold the spicules in position. And then no locomotory organs are present in adults. So the adults are all sedentary. There is no mouth, there is no alimentary canal. Digestion is intracellular. Nutritional mode is holozoic. No excretory, no respiratory organs are present. Nervous system is absent, and response to stimuli is very poor. Asexual reproduction is by budding, by special cell masses which are known as gemmules. Sexual reproduction is by production of eggs and sperms. So there's a free swimming larva which is found in the life cycle, Stereogastrula, which is the larva that is found in the life cycle. We'll come to it later. Sponges have a great capacity for regeneration. So if you take a small piece, by any chance, the sponge gets damaged, it can regenerate again. Skeletons of the sponges have been used commercially for washing, mopping, for bathing, etc., as bath sponge for that matter. So when it comes to classification, so you're going to come to this later, but I'll just tell you flagella, then you have the gastrula, then amphiblastula, invagination to gastrula formation. This is the syncytial hypophysis that is being told, which gives rise to the radial symmetry which occurs in the radiates. So these are the sponges, asymmetrical or radial asymmetrical. You have Pinecocytes, Amiibo Sites, Quinocytes. Central cavity, body wall is made up of Pinecocytes and Pinacodma. So when you see the microscope, you can see the spicule. These are the archaeocytes. Later you will come to this particular slide. In fact, right now let us just check the different kinds of classes. I will be sending both the PowerPoints that I had opened over here, so please stop worrying as to I have not sent. If possible, I'll send it to you a little while later. Okay, otherwise just one of you put in a message in the group. I think that should be enough, but more or less once this class gets over, I will send it to you. All right. What this right, classes of Porifera. So you have class Calcarea, class Hexactinellida, and class Demospongia. Calcarea or Calcispongia. So in this again, as the name Calcarea suggests, the skeleton or the spicules are made up of calcium carbonate. There are no spongin fibers. They are mostly marine sponges. They might be present in hollow or shallow waters. Body is radially symmetrical or vase-shaped, and it has projecting spicules, asexual reproduction by budding. Free swimming larval form, which is a stereogastrula, is present in the life cycle. Example is Leucoselenia, Sycon grantia. And when it comes to the class Hexactinellida, you get to see that these are known as the glass sponges or siliceous lattice or cycinoid forms basically. Skeleton, they are also known as glass sponges. Skeleton is made up of hexactinal siliceous spicules. They are either found separate or in networks. No epidermal cells are present or no mesenchymal matrix. Mesenchymal matrix, whatever if present, it is made up of a union of branching of pseudopodia or the Amiibo Sites basically. And thin dermal layer is present. Mesoglia is thick. Gastral cone is present, which is thin but irregular openings into the spongiocele. Quinocytes are small. Canal system is simple. Budding is by asexual reproduction. Larva is the Stereogastrula. And class Hexactinellida has quite a few economically important sponges such as Euplectella. This is the image of Euplectella. Hyalonema. So this dried skeleton of Euplectella is a marriage gift apparently in Japan. Don't ask me why, because I really don't know the reason why they gift Euplectella. And Hyalonema is a beautiful decorative sponge basically. So yes, when it comes to class Demospongia, most common, abundant in nature, and again 80% of it of the members of Phylum Porifera belong to class Demospongia. Body is very highly organized. It's got spicules which can be tri or tetraxon along with the spongin fibers present. They're brightly colored with amoebocytes with the pigment basically, and their shapes and the colors reflect the habitat that they are present in and the resources that are available. Encrusting on the vertical surfaces or crevices is present, but they have a complicated canal system. Skeleton is made up of spongin and spicules, siliceous spicules. There is fibrocytes which have a high contractile power basically. Canal system is complicated, like I told you. Quinocytes are small. Asexual reproduction is by internal buds or which are actually known as gemmules. And you have a larval form which is known as the Stereogastrula. Example is Spongilla or Cliona. So this is a glass sponge that is present. You have other forms that they have given other classes in this particular PPT. As you can see, you have Sclerospongiae. So again, glass sponges belong to class Hexactinellida in fact. So this is an example of that. Let me see if I can take a picture of Skypha is one. Fucosolinia, which is known as organ pipe sponge. This belongs to class class Calcarea only, in fact. This is Clathrina. Clathrina belongs to the class Demospongia. See such beautiful sponges, in fact. See such a lovely picture, in fact. Glass sponges. This is the Euplectella, which is known as the Venus flower basket. And then, these are the common sponges, the class Demospongia. As you can see, this is Carteri spongia, which is similar to the elephant's ear sponge. This is Spongia, which is known as the bath sponge, commonly used, in fact, for bathing. Now, when it comes to Cycon, which is Skypha, let me see if I can get a picture of Skypha. I think it's there. This is the Skypha, as you can see. So, this belongs to Phylum Porifera, class Calcarea, order Heterocela, family Cytidae, genus Cycon or Skypha. It's a marine sponge found in many different seas, especially in that of the European seas. It's a colonial animal. Each colony has several individuals which are in the form of cylinders. As you can see, this is 1, this is 2, 3, 4, all of them existing together. So in a colony. Again, it's a calcareous sponge. Skeleton has spicules of calcium carbonate. Its body is like that of a flower vase. So it's vase-shaped and it's either gray or light brown in color. Measures around 20 to 25 millimeters in height and 5 to 6 millimeters in diameter. So it's very small. You have a magnified image over here. One end is called as the stolon and the other is free with an osculum over here. Body is radially symmetrical. That means you can cut it from in a radial manner into 2 different halves. Each vertical cylinder of Cycon has a large opening at its free distal end, which is known as an osculum or the excurrent or the excellent pore. This is surrounded by a spicule of monadson calcareous spicules. So the spicules that are present usually are divided into different kinds of based on the number basically. So the spicules are of 4 types: monadson, triaxon, tetraxon, and T-shaped. We'll come to that in some time, but that's what. So this osculum is surrounded by monoxon calcareous spicules. This one prevents the entry of other minute marine animals or large particles into the body cavity. And on the surface, you have minute.
How to differentiate Cycon and Porifera?
Cycon is a member of Porifera, no? What is there to differentiate?
Member.
What is your doubt, babe? You want to differentiate between what?
Part of Cycon is part of Porifera.
Yes, Cycon is a part of Porifera. You have not been listening to the class. It's a marine sponge. It belongs to the class Calcarea or Calcispongae. Okay. So Phylum Porifera has three classes: class Calcarea, class Hexactinellida, and class Demospongia. And we are having a type study of Scypha or Cycon, which belongs to the class Calcarea. Okay.
Okay, ma'am. Thank you.
So, like I said, the water consists on the surface, there are minute pores which are present, which are known as ostia or the incurrent or the inhalent pores. So water keeps entering into the interior of the sponge or inside the sponge body through this pores, in fact. Then it exits through the osculum. So there's a vertical cylinder. Since this is a vertical cylinder, when you cut it into two halves, you can get to see that in the middle there is a cavity which is known as the gastrocele or paragastric cavity. Let me see if I've got a picture of that. Not this slide, the other slide. Right. So these are the monoaxon spicules that are present. These are the other spicules that are present throughout the body. This is the pinacoderm. This is the inside flagellated layer choanoderm that is present. These are the ostia that are present. This is an ostium. See, the pore opens outside and it allows the water to go inside. This is the spongocele or the paragastric cavity or the paragastral or the gastral cavity which is present. It opens out into the osculum. So the spongocele is surrounded by the body wall which consists of the Quinocytes, as you can see. Now, body wall of Cycon has two layers. So it is again diploblastic in nature, that's why. And it has an outer layer of pinacoderm and an inner layer of gastric layer or the gastral layer or the choanoderm. This is the choanoderm. Now, in between, you have this gelatinous layer over here. This is known as the mesenchyme or gelatinous matrix layer. So it's called as the mesenchyme, and it has different types of amoeboid cells which have different functions basically. So again, there are a lot of canals that are present inside the body wall. This helps in the this helps the water to pass through inside the body wall into the spongocele and to exit through the osculum. You have got a very good picture of it in your textbook. Basically, I'm trying to see whether something similar like this. So as you can see, you have dermal ostia which are present, and then this is the transverse or radial canals that are going to be present. And right. If you can see, have a look at the picture in your textbook. I think it will be very helpful. Let us see. This is the filter feeding. I don't want this picture. Right. If you can, you please open your textbook and look at figure 3.5. You can see the Oscula Fringe over there. This is known as the Oscula Fringe. And you have the osculum over here. And then you have the gastric ostia that are present over here. And then you have a spongocele. These are the dermal ostia that are there. And then you have a flagellated chamber. See, this is the flagellated chamber that is present. And then you have the incurrent and the radial canal systems. Basically, the radial canal systems will be something like this. So this is the radial canal system. Inside the body itself, transversely the water can go in. And then you have apopiles. Right. So you have an apopyle, and then you have radial canals basically, like, and then you have triradiate spicules, different kinds of spicules basically. And then you have an amphiblastula, which is the larval form. See, all this, since it is given in a black and white picture in your textbook, not very, very clear to see, but then you'll get an idea of it. And then you have Amiibo Sites present, and then you have ovum, the prosopile, you have a prosopile, and then you have this apopyle over here. This is the prosopile basically. And you have a monoaxon spicules, myocytes, pore membrane ostium, and incurrent canals basically. So canal system is complex. At the same time, it's very, very prominent, and it's the unique feature for this particular phylum, as I already told you. So when it comes to the epidermis, so the entire surface of the sponge or the spongiocele and the incurrent canals is covered by epidermis. So this is the epidermis basically. So you have the pinacocytes. See, these are the pinacocytes. I'm not able to enlarge the picture, but if you can see, these are the pinacocytes. They are flattened cells. Therefore, it's also known as the pinacoderm because the whole layer is made up of that. And they are large polygonal cells. They have a central nucleus. Then the pinacocytes are contractile in nature. That means they can become smaller. They reduce the surface area of the sponge by withdrawing of the margins into the central bulge. So they resemble the epithelial cells of the higher animals. Then there is a gastro layer. This gastro layer lines the gastric cavity and the radial canals. Basically, over here also inside you have the gastro layer that is present. Two different kinds of cells are present. That is the flat epithelial cells, and which are almost like the pinacocytes of the epidermis. And then you have the collar cells, which are known as the quinocytes. So this is the gastro layer. Quinocytes are not present all the time, but if you can actually see over here, you do not have the quinocytes or the collar cells. Collar cells are flagellated, as you can see in the picture, because they have the flagella, they are known as the collar cells. So because of the presence of the quinocytes, this layer is also known as the quinoderm. When it comes to the quinocyte, let me see if I got a good picture of it. Right, this is the cross section of a quinocyte, in fact. So you can see that they represent quinoflagellate protozoans, such as the Proterospongia. Therefore, they are also known as the quinocytes. Cell is oval or rounded, and it has a nucleus and a few contractile vacuoles and food vacuoles present inside it. The apical side is a collar-like transparent protoplasmic outgrowth that is present over here. And a long whip-like flagellum is situated from the base of the collar within the cell, and then it comes outside. Then this projects out. Then beating of the flagella keeps the current of water flowing into the sponge. So that is why you are, and this lines the paragastric cavity and the radial canals also. So when it comes to the mesenchyme, you get to see that this is there in your textbook. Unfortunately, I did not get any kind of slide regarding this basically. So you'll have to just listen to me. In the other PowerPoint also, we do not have this mesenchyme pictures basically. So please listen to me, that's all. So you have polynocytes or connective tissue cells. They are small connective tissue cells which have radiating pseudopodia. They are star or stellate shaped. Then you have chromocytes or pigment cells. So these actually give the pigment granules. So you have to guess by now. This chromocytes means it belongs to the class Desmospongia, where it is colored. Different kind of colored sponges are present. So they impart the color to the sponge. Tissue-cytes are storage cells. Amiibo sites, they are amoebocytes. See, I told you the mesenchyme has different kind of amoeboid cells. So all of these are the amoebocytes present and different kinds of them. So tissue-cytes or storage cells, they are these are amoebocytes or the what do you call it, lobopseudopodia. They have plenty of reserve food granules. Then you have spicules or scleroblasts. Spicules or scleroblasts. they produce the spicules. So you can see that in Cycon, calcareous spicules or calcareous spicules are produced by calcoblasts, siliceous spicules by silicoblasts, spongin fibers by spongioblasts. Phagocytes for food collection, food capture, and they have pseudopodia for the same reason. Trophocytes or nourishing cells. They receive the food particles, digestion takes place through them, and then they transfer the nutrients to different sites of the body. So let me see if I can get one picture of the same thing, the cycon. If I can get a good picture of it, you can see cycon different cells. Now, also I'm not coming now. So as you can see, these are the different kinds. Let's see if I can different types of cells. Some of them have been given over here. You can just see. Like I told you, it's the first multicellular organism, but you do not have a lot of advancement in the cell functioning. So trophocytes are the nourishing cells, in fact, and they receive food particles from the quinocytes, digest the food, transfer the nutrients to other cells of the body. Archaeocytes are the gamete-producing cells, basically, and they are the sex cells. They possess blunt pseudopodia, a large nucleus, and you have a granular cytoplasm, and they also play a key role in the process of regeneration. Lophocytes have a tuft of fibrils at one end. You can see lophocytes. Do we have it in this picture? I don't think it's there. So lophocytes are there. These are their function is not exactly known, basically. Myocytes found around osculum, ostia, apopiles, and other pore connecting cells or canals. They are fusiform contractile cells resembling the muscle cells of the vertebrates. See, H is the myocyte. They regulate the opening and the closure of the osculum. Desmosites. Now, desmosites are mostly found they're at the basement of the canals, basically. They're long and slender. And when it comes to the gland cells, they secrete mucus. This is the gland cells. This is the myocyte. These are the scleroblasts. So spicules are produced by scleroblasts. These are the gland cells found at the base of the sponge, help in adhesion of the sponge to the substratum. They should release some substance to get stuck to the substratum, right? So that is where gland cells play a role. So these are the different types of cells of a sponge. Right. Now, when it comes to the skeleton, I'll go back to our, right. Spicules or skeleton. So various types of spicules are present, either calcium or silica or spongin fibers. So this spongin is a fibrous protein made up of collagen, dried, beaten, and washed, in fact. So when it comes to different kinds of spicules, you have 4 types: monoaxon, triaxon, tetraxon, and T-shaped. Monoaxon are rod, straight-like spicules, and they are found around the osculum. They're known as the mono-radiate spicules, and when they are pair or club-shaped, they are known as the oxioid spicules. So they project on the outer surface of the body and lie outside the radial canals. Triaxon spicules, also called as the triradiate species, they are present along the radiate canals with one ray pointed towards the distal end of the canals. Tetraxon or tetraradiate, so they have 3. See, these are the triaxon, this is the tetraxon, and they occur along with the triradiate canals, and they surround the gastric cavity. T-shaped ones are rarely found. So when it comes to the canal system in the sponges, you can get to see that it is known as the Aquifer System. And sponges are sessile in nature, means they do not have any kind of locomotory organelles, because of which the physiological activity should be conducted only through the water currents. So water enters through the ostia, exits through the osculum. Excuse me. She should. Chalo, go. Take the phone and go out. Right. So the canal cyclone consists of incurrent, radial, and excurrent canals. They have an opening ostium and oscula, or ostia and osculum, in fact, different kinds of ostia and osculum. And then you have intercanal apertures such as the prosopile and the apopile. So these are intercanal apertures. Let me see if I got that picture again. Getting a good picture is so difficult sometimes. You just have to keep on searching. Still, you'll not get a good picture, unfortunately. Anyway, so as you have already seen, read about it. So this is the Pinecocyte or site. These are the porocytes. This is the pore or the ostium, basically. Quinocytes. These are the flagella. These are the sclerocytes, archaeocytes, spicules. This is the mesenchyme, which is made up of gel-like substance. Okay. Trying to see one picture, good picture with a water vascular system, in fact. Sorry, it's the water canal system. So you have prosopile and apopile also present. Please have a look at your picture that is there. Then the one that is there in your textbook. So that should also help you basically. Right, I'm sharing my screen again with you. Something which gives you a basic idea. Okay, so as you can see, you have an incurrent canal. This is the incurrent canal. This is the incurrent canal. This is lined with flattened pinecocytes. Outer end of the canal is slightly dilated basically, and it terminally ends into the paragastric cavity. Now a thin membrane which has 2 or 3 minute ostia is present over the outer dilation. Now you have radial canals over here. Okay, over here you have the radial canals. So this radial canal, it's also known as a flagellated canal because of the presence of flagella. Then on the inner side of it, it is connected with the paragastric cavity. I do not know if you've seen, this is the paragastric cavity. On the inner side of the radial canal, it is connected with the paragastric cavity. Outer end, it just terminates blindly. There is no, as such, any kind of outer end. And this has the radial and the incurrent canals are interconnected by short, narrow channels which are known as prosopiles. So these are the prosopiles over here that you can see, and each prosopile is a perforation in a single tubular cell which is known as a porocyte. Now When it comes to the excurrent canal is where you have a short wide passage through which the flat ectodermal cells, that is the pinacocytes, the radial canals open into the paragastric cavity. So this is the incurrent canal, this is the excurrent canal basically. So it opens into the paragastric cavity. Now when it comes to the course of water current, you can get to see that first water enters into the dermal ostia, the water current through the water current, then into the incurrent canals, from into the prosopiles, from the prosopiles into the radial canal, into the apopiles, then to the excurrent canal, then to the spongoxyle or the paragastric cavity, then to the osculum, then to the exterior. So this can happen as a cycle. So when it comes to the advantages of the canal system, you get to see that it is a very significant feature in the sponges. So the continuous and uninterrupted flow of water current is very important for sponges, and the water current helps in bringing food, helps in bringing water, oxygen, and it helps in the sperms and the egg to move out of the body, removes the nitrogenous wastes and carbon dioxide. Amphiblastula larva escapes from the parental sponge through the canal system, helps in the dispersal of the species. And it's estimated that in a sponge which is around 10 centimeters long or 1 centimeter wide, I'm talking about this size, and 1 centimeter width, there are about 22 lakhs 50,000 radial canals present or flagellated chambers present. This pumps around 22.5 liters of water per day through its body. When I'm talking about 10 centimeters, it is just this much, as long as a small scale that is present. So, and it pumps around 22.5 liters per day, it seems. So that's the kind of sponges water canal system that you can see and how important it is. So I'm going to stop sharing this particular one. Let me see if I can go back to the PowerPoint. So when it comes to asexual and sexual reproduction, you get to see that asexual reproduction takes place by budding. Sexual reproduction takes place by takes place by the sex cells. So can you please mute yourself, whoever it is? I request you to mute yourselves. So let me share my screen with you again. Perfect. Right. So when it comes to the asexual reproduction, it is by budding or regeneration. Regeneration occurs when the body part is lost due to injury, and budding is a common method for asexual reproduction. Takes place, and this takes place by external budding. Masses of archaeocytes, which produce the sex cells, they migrate, form an outgrowth called as a bud. This rises from the base of the sponge, acquires an osculum at the top, then it either becomes free or it remains attached to the parent sponge, and then it adds to the size. So that's how budding takes place. So when it comes to sexual reproduction, you can get to see that most of them have a cycon. Cycon is a hermaphrodite, meaning that both the male and the female organs are present in the same animal. And this is done by the sex cells, which are the sperms and the ova. And the cycon is a protogynous creature, meaning that female gametes mature earlier than the male gametes. So special sex cells, which are, or the testes or the ovary, are all absent. We are just talking about the most primitive multicellular organism ever present. Sex cells arise from special Amiibo sites, which are known as the archaeocytes. Now, when it comes to the sperms, the sperm mother cells or the spermatogonia, they come from the Amiibo sites. They repeatedly divide. Once they repeatedly divide, then what happens is they get a cover basically, and they divide 2 or 3 times to form the spermatocytes. They again divide into, they become sperms after a meiotic division. Now, a mature sperm has a small, a long, vibrant tail, and it has an oval or rounded or a pear-shaped nucleated head. And along with the water current, they are carried out into the flagellated chamber of another cycon. So to go into another cycon, what has to happen? This particular one cycon has to release the sperm into the osculum, and through the osculum, it has to go travel, and then it has to go out into the sea. Then it has to go to the next cycon, which is present. It can be present to the parent itself, or it can be present somewhere else. So this is how reproduction takes place. So when it comes to the ova, the egg mother cells are amoeboid, wander through the mesenchyme to the base of the quinocyte layer in search of nutritive cells. Now they increase their size by receiving the nourishment from the nutritive cells. Then they undergo meiotic division to develop into ova. Right? Now, what happens is they lie in the wall of the radial canal there. Now, when it comes to the fertilization, the union of the egg and the sperm, that is the male and the female gamete and the male gamete, is known as fertilization, and this forms a zygote. Now, the fertilization in the sponges is internal. Mature sperms escape outside in through the current of the water. They enter the body of another sponge along with the incoming water current. Then they reach the radial canals. In the radial canals, you have the ova present, right? So here the sperm enters a wandering amoebocyte or a quinocyte, which is called as the carrier cell. It loses its tail. It gets internalized by these cells. Now this wandering quinocyte will carry the sperm to the oocyte. Then the sperm penetrates, fuses with the egg, completing the fertilization. It results in the formation of a zygote, which gets enclosed in a brood capsule formed by the lining of the neighboring amoebocytes. All this is taking place in the wall of the radial canal. Remember. So let us look at the development. So we have the development of this is a good picture that I get. Right. So development is a series of events that take place or occurs in the organism during the transformation from the fertilized egg into the adult animal. So in all the metazoan animals, it commences with the division of the zygote. Now, once the zygote, then it divides into different planes at the right angles to one another, forming a geometrical ratio, forming cells in a geometrical ratio. That is 2, 4, 6, 8, 10. I'm so sorry. Not 2, 4, 6, 8, 10, 12. It is 2, 4, 8, 16. So what is happening is the number is doubling every time. 2, 4, 6, 8, 10, 12 is skip counting. I was teaching my first standard, son basically. So I was like 2, 4, 6, 8, 8. No. It is 2, 4, 8, 16, 32, 64. So the cell count or the cell number doubles with each division. Early stages of development in the cycon occur in the radial canal of the mother sponge. So when it comes to the cleavage, zygote undergoes a holoblastic cleavage. First 3 divisions are vertical. They produce 8 cells or 8 blastomeres. This is followed by a horizontal division. What vertical means? 1, 2, 3, 2 cells. 2 cells, so 4 cells. to 8 cells, and then you have a horizontal. So this is the 16 cell stage that you're able to see in the on your screen right now. So of these 16 cells, 8 are lower. They are known as the macromeres, and 8 are upper and smaller. These are known as the micromeres. The macromeres form the dermal layer, and the micromeres form the quinocytes. So the embryo now appears like a flattened disc, and the cavity appears, which is known as the blastocele. A small cavity appears. So this is known as the blastula stage. Now you have the stromoblastula stage. Excuse me. So in the stromoblastula stage, the small micromeres, whatever they are there, they multiply rapidly, become elongated, they start getting flagella. Now the flagella are present on the inner side facing the blastocele, as you can see. The macromeres do not divide, but they become granular. These are the macromeres. They do not divide, but they become granular. Now, this stromoblastula, in the center of the macromeres, an opening appears that functions as a mouth. Embryo ingests cells from the parental sponge. This embryonic stage is called as the stereogastrostomoblastura. This embryo is essentially blastula, but with internally directed flagella and a closed mouth. Now, it undergoes inversion basically, and then you get the amphiblastula stage, wherein, over here, the stromoblastula stage, basically, the macromeres are down, but then over here, after inversion, the quinocytes and the quinoderm layer or the micromeres come out. It undergoes this particular shape basically. So the embryo now undergoes a remarkable process of inversion, which is not seen in any other metazoans. So the mouth gets reopened and to expose the flagellated cells, which are directed downwards now. Now the macromeres slowly multiply, spread on all the sides. Then the mouth of the embryo closes and the embryo becomes elongated. And the embryo becomes elongated. So, see, as you see over here, this is what is happening here. So, this free-swimming flagellated embryonic stage is actually known as the amphiblastula, and it is externally and internally different from the immobile adult. So, until this stage, the embryo will be in the mesenchyme in the brood capsule formed by the quinocytes of the parental sponge. Now the brood capsules rupture, embryo gets detached, enters the radial canal, finally comes out of the parent body along with the outgoing water current, swims actively, becomes the and marks the ciliated larval stage of the life cycle, that is cycle. Now gastrulation is when the amphiblastula larva now swims for some time with the flagellar end directed forward. Then what happens is this flagellar cells invaginate, and then they are overgrown by a large number of granular cells. Now embryo becomes two-layered. So as you can see, this is the gastrulation stage, wherein embryo becomes two-layered. You have the outer pinacoderm or non-flagellated cells. Inner is the quinoderm or the flagellated cells. So this is the diploblastic larva, which is known as the gastrula. You can see that it's there in your textbook basically. So when it comes to the metamorphosis, you get to see that it is a phenomenon of structural changes which is necessary for the transformation of the larva to the adult form, and especially in animals where the larva is morphologically and anatomically different from that of the adult. So gastrula larva elongates, becomes cylindrical, then there's this osculum which is getting developed at the free distal end. Then small perforations start appearing in the cylinder, they become the ostia. Then you have the Pinecocytes and the Quinocytes and the various Amiibo Sites that are getting developed. This stage resembles a simple leucocorelia-like sponge, and this is known as the Olympus Stage. This develops into an adult cycle. Now, I told you already that the canal system in Porifera is very important for all the reasons, be it for water, bringing the food particles or for oxygen or removal of metabolic wastes or for reproduction, dispersion of species, etc. So it's a complex one. As the classes are going on, as you get to see, the Demospongia has the most complex canal system present. Different kinds are there, especially three of them are there, which are main basically, which are Ascon type, Sycon type, and Leucon type of sponge systems. So to put it simply, this is the asconoid type or the Ascon type of canal system, basically simplest one, and found in ancient sponges. And you can see that there are incurrent pores or the ostia that extend from the external body surface. They go into the spongocoel, out of which each pore is actually intracellular, and that is a canal through a tubular cell called as a porocyte. Water passes through the ostia into the spongocoel and through the osculum to the outside. So this is as simple as it gets. And so as you can see, the dermal pores, ostia to spongocoel to osculum. It is slow, not very efficient, as the gastrocoel has a large amount of water that can and cannot be circulated always and replenished rapidly. As a result, these sponges are very small in size and as compared to the other advanced forms. We have the Sycon type of body wall, that is the Syconoid type. In this, what happens is, it is labeled actually in your textbook. If you can find a picture of it, it is very good. Follow your textbook. You can see this picture also. This is the Sycon type. Over here again, what happens is, water enters through the dermal pores because they are the openings of the incurrent canals. Basically, these are the dermal pores. Then they open to the radial canals. Radial canals lead the water to the spongocoel again. See, this is also the spongocoel. The radial canals that are present will lead the water to the spongocoel, and quinocytes line the radial canals as well. So this beating of the flagella moves the water through the from the ostia through the incurrent and the radial canals to spongocoel and out through the osculum. So over here, what kind of water canal system that is present is, it follows the route of dermal pores. Dermal pores through the spongocoel, basically the incurrent canals. The incurrent canals to the prosopiles that are present. Then the radial canals, then the apopiles to the excurrent canals, to the gastric ostia that are present, the spongocoel and the osculum. So this is all about the Sycon type of body system. Then we have the leuconoid or the Leucon type of canal system. It is the most evolved and the most complicated water canal system that is there. Water enters into the osculum, moves through the branched incurrent canals. Then they lead to the Quinocyte-lined chambers. Then canals leading away from the chambers, they are actually known as the excurrent canals. And then from there you have water, there's a proliferation of the chambers that are present and the canals. So you do not have a spongocoel as such, which is present basically. So the quinocyte layer of the radial canal of the leuconoid stage evaginates into many small stages. Repeatedly, what happens is, so repeatedly it is filled with mesenchyme around the flagellated chambers. Then the wall of the spongocoel becomes folded for the second time so that excurrent canals lined by the Pinecocytes are present. Kind of complex if you see. Now, the excurrent and the incurrent canals further get branched. Spongocoel is more or less very obliterated, meaning it is also, it's all over. You do not have a particular solid spongocoel as such. It is all diffuse or obliterated. So what happens is, there is an indefinite form as such. So radial symmetry is lost in this leuconoid type of canal system animals. Surface is covered with epidermal epithelium pierced by dermal pores, which are ostia. and Oscula, and these lead into the incurrent canals. And then there is this irregular mesenchyme throughout, in fact. And so you have small openings with the flagella. They are known as the prosopiles. And then the flagellated openings open by apertures called apopiles into the excurrent system. Let me see if I can get a picture. Right. So as you can see, you have an incurrent or incurrent canal. You have the apopiles, which lead water to the outgoing or the osculum basically. And you have the dermal ostia over here. This is the incurrent canal leading to the prosopile basically. And from the prosopile, the flagellated chamber to the excurrent canal through the apopile to the what do you call it, osculum. So if you excurrent canals, I mean, sorry. So again, there are different kind of variations in the Lupon canal type of canal system. In fact, they are known as the Europhilus, aphodal, and diplodal. Europhilus, in this type, you have wide thimble-shaped flagellated chambers that open directly by wide apertures, which are called as apopiles, into the, what do you call it, into the excurrent canal. So apopile opens into the excurrent canal. Just please remember this. Apopile opens into the excurrent canals, and they receive the water supply from the incurrent canals from the prosopile. So prosopile receives water from the incurrent canals. Apopiles receive the water from the excurrent canals. Aphodal means in many of the leuconoid sponges, what happens is apopile of each flagellated chamber, whatever is there, this is the apopile, is drawn throughout as a narrow canal called aphodus. Therefore, it's known as an aphodal system. Examples are stellata and geodia. Examples of Europhilus are Placotis and Tetula. And diplodal is the third type, wherein besides the aphodus, another narrow, minute canalicule is present, which we called as the prosodus. If present, it is between the flagellated chamber and the incurrent. So this is the incurrent canal. So incurrent canal and a flagellated chamber. This is also another incurrent canal. This is the flagellated chamber. If possible, this is a prosodus. So when the prosodus is present, it is known as the diplodal condition. Example is chondrosina and spongina. So the course of water current, if you see, it is the dermal ostia. These are the dermal ostia to subdermal spaces to incurrent canals to prosodus if it is present, to the prosopile, from the prosopile to the flagellated chamber, from the flagellated chambers to the apopiles, and from the apopiles to aphodus if it's present, and then through the excurrent canals to the large canals to the osculum. So this is the course of water current. Now, there is also another last one, which is known as the Ragon type of canal system. In this, the calcareous sponges, the leuconoid structure may arise by a way of asconoid and cytinoid stages. But in class Demospongia, you have the Ragon type. Let me just check if I take a picture of it. Right. So more or less, this is the Ragon type of canal system. Not a very good picture, unfortunately, but please adjust with it. So, right. So in this again, if you see the kind of water current that is going on in the Ragon type is, it's a flattened or pyramidal like conical sponge, tapers from the base to the apex. And on the apex is situated the osculum. Basal wall is called as the hypophare, and it is devoid of any kind of flagellated chambers. Upper wall has a small row of oval flagellated chambers. It's called as the spongophare. Spongocele is bordered by oval flagellated chambers opening into it by wide apopiles. Mesenchyme is situated between the flagellated chambers and the epidermis, and incurrent and excurrent canals may be complex or branched. So the course of the water current in this Raygon type is ostia to subdermal spaces, to incurrent canal, to prosopiles, to flagellated chambers, to apopiles, to excurrent canals, to spongocele, and then the osculum. So this is all about the Raygon type of canal system. So if you actually see, when we summarize it, Phylum Porifera is comprised of sponges. They are multicellular sessile animals. Body is with many pores, which are meant for the passage of water. Performs various kinds of body functions. Skeleton is made up of spicules. Different physiological functions are carried out, and this is all in collaboration with the water currents. And then reproduction is quite both by asexual as well as sexual reproduction. Asexual reproduction is by regeneration or budding. Sexual reproduction is by the union, or the fertilization is by production of sperm and egg cells. And then you have the the larval form, Stereogastrula, is basically mobile in nature, whereas the adult is a sessile calcareous marine sponges, or sessile in fact. Then they are diploblastic. If you come to Cycon, that is, it is a sessile. That means it does not have any kind of locomotory organelles. Calcareous marine sponge, it is diploblastic in nature, 2 layers, outer pinacoderm, inner choanoderm. Then physiological activities are accomplished by current of water flowing through the system of canals. Asexual reproduction by generation or regeneration, budding. Sexual by through sperms and eggs. Fertilization is internal, development is in situ at the same place. That means free swimming larval form occurs. And then a canal system or in the Phylum Porifera reveals the complexity because of the evolution of these organisms or animals. And this helps in the efficient and the speedy replenishment, that is usage of the water that is there in the canal system. So this is all about your Phylum Porifera. Next class, I'll talk about the phylum Nidaria, their general characteristics or general characters, classification, type study, Aurelia. We have Aurelia, that is the jellyfish, polymorphism in Nidarians. So this is all for today, basically. And have a safe, please stay safe. Is there any doubts as of now?
No, madam.
So yes, like I said, I will send you, what do you call it? Sir, I'm ending the class now. Anyway, I'll just send the PPTs to you. Yesterday's and today's. Okay, thank you.
As of now,
Some important topics.
Important topics you'll have to ask in the university, madam. And I will be ending the class right now. It's already 5:10. 10 minutes past your class time being basically. Yes, I will send you your PPTs, all these 3. They just and these are only for your reference. Please follow your textbook religiously, whatever it is. Okay?
okay, ma'am. Okay, ma'am.
Yeah, thank you so much.
Yeah.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, madam.
Thank you, ma'am.